Now, up to this moment, I think every one of you are very familiar with this kind of monohybrid inheritance that Mendel's proposed. And at that time, even though when Mendel first proposed the idea, not much people are convinced, but as time goes by, more and more people start to find that this principle makes sense and it explains quite a lot of phenomenon. However, this is going to change, and in 1908 by a scientist called Thomas Morgan Hunt, and he's going to discover a new way of inheritance that Mendel never think of and never find out. And Morgan's story is one of my favorite, so I hope to show you, like how I show you Mendel. I hope to show you how Morgan discover the new idea, and we're going to go through his investigative process and think like Morgan and see whether you, when you look at the results of those breeding experiments, I hope you can also discover what Morgan find out, and I hope you enjoy the process and learn the new way of inheritance, which is actually quite complicated when you start learning it. But over time, of course, you'll be okay. So the story comes to the time where, if you remember, Mendel's proposed the law of inheritance at a very early time, and then there is no physical evidence until Walter Sutton, the 1-year guide mentioned by Elaine, really observe chromosomes, and that time people start to believe that the inheritance factor that Mendel's proposed is present in the chromosome, and we are talking about the theory of chromosome in inheritance. That means chromosome plays an important role. The genes is present on the chromosomes. There is a scientist called Thomas Morgan Hunt, which doubted that at the start. So he, being a geneticist, studies the inheritance by using fruit flies, because like pea plants, fruit flies grow very quickly and grow very cheaply. So it's a good model organism for studying a lot of generations and a lot of offsprings, and you can get reliable results in crossing. So what Morgan's lab doing is to study various phenotypes in a fruit fly, and he grow a lot of fruit flies, and he hope to find new varieties, new phenotypes of in the fruit fly that no other scientists have discovered. And after many years of growing fruit flies and observing them under microscopes, on one day in 1908, after long time of waiting, he finally obtained a mutated fruit fly that has white eye. So the next picture shows you how it looks like. Amazing, right? So here's the first scientist to discover or obtain the fruit fly with white eye. Then the next question he posed is that, alright, so how would this new phenotype be inherited along generation? Because no scientists, no geneticists have studied that. So, as usual, like what Mendel did, he crossed with the white eye male with other fruit flies. So we're going to study these crosses one by one. and you'll see how amazing it is. So you can refer to your worksheet if you can't see the crosses here clearly, but please follow the sequence that I'm going to guide you through, and this is how Morgan think in sequence. Well, since he get 1001隻 white eye male, and luckily it didn't die quickly, so the first thing that he did is to cross it with the female that he has, which is all of the red eye. And of course, the red eye ones don't have that rare mutation at all, because it's so rare. So we are first sure that those red eye females are pure breeding, homozygous. Okay, so when crossing this white eye male with the red eye female, all the offsprings have red eye. What does it tell you? Obviously, it tells you that the red eye is the dominant phenotype. Now up to this moment, everything is so Mendel, right? Mendel did that too. Nothing surprising. So what did Mendel do next? Right, so Morgan did the same thing as Mendel. He crossed this red eye F1 with another red eye F1. Alright? Do you remember Mendel's result? Yeah, I hope you remember that the F2 that he obtained, is it red eye or white eye or both? What's the ratio of red eye to white eye? So both phenotypes exist in Mendel's case, and the ratio is 3:1. Look at cross B then. The red eye of one that Morgan obtained, he crossed the male one with the female one. The result here is that the offspring has both red eye and white eye. The ratio of red eye offspring to white eye offspring is 3:1. Up to this point, is it surprising? Does it follow Mendel's story? Now look at the results carefully in cross A and B and answer question 1. Consider Morgan's cross A and B. Did the results differ from traditional Mendelian genetics? What's surprising? So please use 2 minutes to look at that and see whether you can identify the surprise in Morgan's results and how that differs from Mendelian genetics. "30 seconds up." I think most of you get the idea how the results differ. I want you to think carefully. How about Mendelian genetics? How would that result be in such cases of the F1 self-crossing? Still 3:1, right? The 3:1 is not something special. It is the gender, the sex of the fruit fly that is quite surprising. So, how did the results of Morgan's experiment differ from Mendel's one? I think everybody can feel that this is something special. All the white-eyed fruit flies are male, and there are no female haplotypes with white eye. This is very surprising and totally new, because if you look at the traditional Mendelian cross, we never care about the sexes, right? So if this is a brown-eyed mother, this is a brown-eyed father, let's say, the 3-to-1, these can be daughters, these can be sons. The blue-eyed ones, these can be daughters, these can be sons. But in Morgan's experiment, the only male F2 have white eye. Now you see the surprising results. Did you have any idea? I mean, what idea comes to your mind? From this result, Morgan had an idea. He proposed that the eye color seems to be inherited in a Mendel's way, except one thing. It seems that the white eye feature is somewhat linked up, or even you can say locked up, with male, with the feature of male. That means the white eye, or in general, the inheritance of eye color and the inheritance of sex seems to be related, or in his term, seems to be linked. And he thinks that it is this linkage that causes the white eye to be only male. Now, to further test his hypothesis whether the inheritance of eye color is closely linked up with the inheritance of sex, he followed up his idea. He followed up with an amazing experiment. He did a very simple thing. Eventually, he obtained a white-eyed female after many crosses. He did it in a crazy way. And he crossed this white-eyed female with a red-eyed male, which is quite sure to be purebred. Look at the results. The results is that both red-eye and white-eye offspring comes up, and all the female have red eye, and all the male have white eye. Now, based on cross C, does it give you the idea about sex linkage? How does the result differ from Mendelian genetics? So look at question 2 in this worksheet. So consider both cross A and C. What are the amazing things that makes it different from Mendelian genetics? I think there are 2 surprising outcomes. So again, 2 minutes, see whether you can understand, and during the time I'll put some probing to guide you think. Now again, look at cross A and C. Okay? So cross C alone give you one surprising result compared with Mendelian genetics. If there is a purebred red eye, purebred white eye, what Mendel expect the offspring to be? Expect them to be all red eye, right? Like A. So having both red eye and white eye in the offsprings is already an amazing result, which differs from Mendel's expectation. But if you look at A and C at the same time, there is one more amazing thing, particularly if you look at the parents first, I would say the cross A and C share some similarity. What's it? What is it? In terms of phenotype, is there any similarity? Yeah, it's both red cross white. The only difference is that the sex is switched. So, right? Now if you go back to Mendelian genetics, let's say this is father, this is mother, and you get a 3-to-1 ratio. Imagine now you switch the sex, this becomes the father, this becomes the mother. Will the result change? Not at all, right? Because no matter it's a father or mother, we always write the big A, small a. We don't care, right? So, for the same phenotype, no matter how you switch the sexes in Mendelian genetics, the result is always 3-to-1. At least always the same, right? This Morgan's experiment gave him a very, very big hint that the eye color and sex is closely linked in their inheritance, because with the same parental phenotypes, when the sexes is switched or swapped, the outcome the outcome is completely different. Now appreciate this and compare that with Mendelian genetics. This kind of this is a big signal. Whenever you see something like this in a question, in exam, or in a real cross, it gives you a very obvious signal that this inheritance is closely linked with sex. So, without doing any detailed analysis yet, you already see that from cross A, B, and C, and particularly B and C, in cross B, only males have white eye. No female has white eye. In cross C, when the sex of the parents are switched from cross A, it affects the results. Now, these two features does not exist in any Mendelian stories at that time. So Morgan has to find a way to explain these kinds of results. So please spend 2 to 3 minutes, finish up the page 1 of the worksheet about question 1 and 2, give you some time to sum up the ideas we have just discussed, and then we are going to end like what we did last time during the holiday. Morgan tried to use a model to explain these outcomes. Alright, so Morgan knows that. So Morgan put his focus in this white eye male. At that time, they know how sex is determined, just like we have discussed in the previous section, and the sex determination in fruit fly is the same as the sex determination in human. XY chromosome, male being XY, female being XX. So the key idea that Morgan comes up with is that, alright, this is a white eye male. Its X chromosome must come from his mother. So he thinks that, alright, so this eye color thing may be somehow located on the X chromosome. He coined the word sex linkage to describe this idea that the gene controlling a certain characteristics is linked with sex. And the reason that they are linked is because the gene is exactly located on an X chromosome. So he tried, now a hypothesis come up, right? To check whether he is correct or wrong, he need to use this idea to explain all three crosses. And if it can explain all three of them, then his idea seems to be a pass. So we are going to help him, or we are going to reenact his model and see how Morgan used his hypothesis to explain these three crosses one by one. So that's the page 2 of your worksheet. So let's do these one by one. I'll do the first one as an example for you. I hope you learn the format and the way of thinking, and then I hope you can do the cross 2 and 3, or cross B and Z on your own. Like every genetic diagram, we need to define the symbol. So this is big letter R represents the allele for red eye. The small letter R represents the allele for white eye. Now, I don't write yet because I'm going to write something wrong first. If I write the cross one like this, okay, it doesn't show the idea. This is wrong. Can you think about why? Remember that, I'll give you a hint. Try to find out what's wrong in this description of genotype. Morgan suggests that this gene controlling eye color is located on the X chromosome. Which genotype here is wrong? "30 seconds. If you know which one's wrong, try to explain to your partner why. So which genotype is wrong? The female one or the male one? The hint is Morgan is now proposing that the allele is on X chromosome. Alright, so I hope you can successfully find out that this is wrong. The male one is wrong because male only has one X chromosome. So the genotype should not have two alleles for white eye color. Instead, it should only have one. So it should only the genotype for the male should only consist of one small r. But if we write like this, it doesn't show the feeling that this allele is so special because it is on the X chromosome. So when somebody see that you say this genotype is one single letter R, they don't understand why, and there's no way to explain to them so much, right? So we will change our way of presenting these genotypes. Still, we let big letter R to be allele for red eye, small r to be the allele for white eye. We're going to draw the parent phenotype, which is a red eye. And now this time the sex is important, so we have to write the sex as well. White eye, male. So the first thing that these genetic diagram differ from the traditional one is that you must show the sex in the phenotypes. The second thing that differs is that we want to show or emphasize that these alleles are present on X chromosome. So we are going to present the genotype as XR, XR. the letter X, we don't define it because we don't need to. X stands for X chromosome. So when I write the symbol XR, it represents that the R allele is located on X chromosome. So remember, don't let XR be the allele for red eye. This is wrong, because X is a chromosome, R is the allele. The symbol X, big letter R on top, means that this allele is located on X chromosome. So now, can you write the genotype for the white eye male? Sorry. It should be, first think about that a male has an XY, one X chromosome, one Y chromosome. How many we put the small letter R here because it's on the X chromosome. So how about Y? We don't need to put anything on it because if you remember, XY chromosome contains different genes. So there is no eye color gene at all on the Y chromosome. After that, Morgan used Mendel's idea in the law of inheritance. He thinks that, alright, so the allele here separates during gamete formation. Each gamete only receives one allele. So here there are two possibilities, X more R or Y. So after fertilization, let's see how the F1 looks like. Now, one type of the F1 consists of X di R, X di R. It is a red eye female, while the other one is X di R Y. So this R would be expressed because it's dominant, but in another concept, there's nothing to compete with it because there's no eye color gene at all on Y chromosome. So it must be red eye again. And this is a male. So therefore, at first sight, when Morgan didn't care about the sex in cross A, all offsprings are red eye. So that's the explanation for cross A. Now let's see how amazing cross B is. Particularly, we would see whether Morgan's idea can help us explain why the only male would be white eye and no female would be. So I'll give you 2 minutes to draw the genetic diagram on your own. Remember that this F1 is the F1 from cross A. So don't make a mistake there. if you're finished, try to I'll give you 1 more minute, okay? I'll give you 1 more minute. For this minute, please finish up the drawing, but more importantly, spend some time to feel, or try to even orally explain how come there is no white-eye female. Alright, at the meantime, I'll draw my own diagram, but I hope you would do that is explain how come there is no white-eye female, and try to see whether Morgan's proposal matches with Crosby or not. Sorry. Alright, so let's check the answer. Alright, that's good. Is it alright? Okay. So let's check the answer and see how amazingly there's no white-eye female. This is the key. Alright. Let's see. The red-eye female here comes from the F1. It's a hybrid, so it's XIR. The red-eye male, since it has red eye, of course, is X big R, and being a male, the other sex chromosome is Y, with no R on top. Now, the gametes X big R or X more R, X big R or Y. Fertilization takes place, so this X big R, X big RY, X big R, X small r, and the X small r Y, which is so special because this is now the white eye male. Don't just write these to be red eye, because we want to talk about sex. So you gotta say this is a red eye female, this is a red eye male, this is a red eye female. So therefore, if I only consider the red eye to right eye ratio, it's still 3:1, Mendel's ratio, right? But the amazing thing is that there is no female. Both female have red eye, and the only white eye can only be present in male. I want you to answer a question like this. So, 1 minute. In the picture, in this genetic diagram, can you circle the key allele, gametes, whatever, that contributes to the fact that no female would have white eye? Which allele or which gamete or which X chromosome, whatever you want to talk about, which X chromosome, let's say, or Y chromosome, contributes to the fact that there's no white eye female? Obviously, is this one. Is the Now, in real exam, of course, you don't circle, alright? But here, the amazing point is here. This male red eye, this X big R, must be delivered to the daughters, right? Because otherwise, if it delivers the Y, it must be male. So this XR must reach every female offspring. It makes them all having red eye. Next question. How about the white eye male? Which chromosome makes it to be white eye? Of course, it is this X more R, and this X chromosome is inherited from the heterozygous female. Okay, so I leave the homework for you as for cross C. So go home, or during the lesson time now, please draw the genetic diagram for cross C and see how it works. But before you do that, I'll give you free time for that during the video. But during you doing that, let's finish up the story. So now Morgan has an idea. Some phenotype, like the eye color in the fruit fly in this case, is inherited differently from what Mendel suggests. The key difference is that this phenotype is linked with sex inheritance. He called these type of phenotypes sex linkage. And the definition is that the factor, or nowadays we call gene, the gene that is present, controlling a sex-linked phenotype must be located on sex chromosome, and in this case, X chromosome. So, go home and do this, alright? And we'll come back to the notes first. So, we call a gene sex-linked when this gene is located on sex chromosome. And in most of the case, the sex-linked genes are X-linked, meaning that they are located on X chromosome. Does it make sense? I mean, does it make sense that most of the sex-linked chromosome genes are on X chromosome rather than Y? It makes sense because X chromosome is much longer than the Y. Now, of course, there are Y-linked genes and alleles, but to be honest, in DSE, most of the cases will be X-linked. But don't, again, follow the data during exam. So, there are some more examples in human that, so the Morgan's idea is in fruit fly, but humans' story is in sex, is of different phenotypes. So there are 3 common phenotypes that is sex-linked. We are going to talk about color vision here. The other 2, please find the textbook to find it out and put it here. So here there is a story. The color blindness is a sex-linked recessive character. A colorblind husband and a heterozygous normal wife are going to have a baby. Is it a must to tell you that a normal female is heterozygous? Is it a must? Or can I just say normal wife? Think about this as a recessive disease. So a normal female can be either homozygous or heterozygous. So I gotta stress that is a heterozygous one. And learn a word, we call when there's a disease, okay, we call a heterozygous individual a carrier. Because it carries what? It carries the diseased allele, but it doesn't show the disease. So that's why we call it carrier. So very quickly, we'll draw this gene, or let me say, I'm confident in you. Can you please draw the genetic diagram by yourself? I'll give 5 minutes to finish this one and the previous one in the worksheet, and we are going to proceed to the next worksheet, which is the most you can say that the most important one in sex linkage story. Okay, the 5 minutes.